Good evening, guys, and today we will discuss the Pathoma book of pathology, and I will make series of the lectures, topic by topic and chapter by chapter. So stay tuned. Today we'll start the first chapter, the first topic: growth adaptation, cellular injury, and cell death. Our topic is growth adaptation. It means that the idea here is that when stress is put on a cell or organ, it induces changes in it, and it causes growth adaptations. For example, if the stress is increased, there are two terms which are used interlinked. These are hyperplasia and hypertrophy. um, if the stress to an organ is increased, for example trauma or excess something, it causes an increase in the organ size. Okay, it is directly proportional to the changing the organ size. If the stress is increased, the organ size will automatically will be increased. For example, the man who goes to the gym, so they uses their muscles extraordinary, and their muscle size automatically increases. It's the case in which the stress is increased. There are two types. Two kinds in which the organ size can be increased. First is the hypertrophy, and second is the hyperplasia. Hypertrophy means that the cell size is increased. The size of the cells is increased. Hyperplasia is number of the cell is increased in a particular organ. The phenomena underlying the gene activation is the central dogma. Central dogma in which the gene is expressed, and it is transcribed into RNA, which is translated into proteins. This is the synthesis of the protein ultimately from the gene activation, and organelles production, increasing in the number of organelles. Okay, as far as we are concerning the increase in the size, so we need to increase it in these particular ways: gene activation, protein synthesis, and organelle production. Okay, the number of cells is increased via one mechanism, which is the production of production of new cells from the stem cells, like from the progenitor cells, which are included in the stem cell of a particular cell. For example, growth lineages. For example, RBCs are produced by the erythroid progenitor cells, so that production is increased in the stem cells. Both the hypertrophy and hyperplasia are seen together. They are seen together in the uterus during pregnancy, and it is physiological, it is not pathological, because when an embryo is developing inside the mother's uterus, so the uterus size, the cells of the uterus, they also increase in the size, and the number of cells also increase as to carry the baby during the gestation period. Okay, the important point here is that we all know that permanent tissues, there are various permanent tissues in the body. For example, the cardiac cells, nerve cells, and skeletal myocytes. These are the cells, they do not undergo hyperplasia. They do not undergo hyperplasia, in which the number of cells do not increase. Only their size can be increased. They undergo only hypertrophy. Cardiomyocytes, for example, if there is hypertension, or like here example is written, if there is systemic hypertension in any person or any patient, so the left ventricle particularly has to do more work, but it only leads to hypertrophy of the left ventricle. Like in the diagram here, it is mentioned that during the systemic hypertension, the cardiomyocytes of the left ventricle, this is the left ventricle, they increase in size. They increase only in size. As microscopic examination is concerned, they are not increasing in number. Other like neurons, nerve cells, skeletal muscles, they increase in size, not in the number. Okay. This one is the physiological hyperplasia. Okay, pathological hyperplasia means in response to any diseased condition. For example, endometrial hyperplasia. Endometrial hyperplasia is a condition in which the endometrium of the uterus increase in the size because the number of the cells inside that is increased. One point here is worth mentioning that estrogen is a hormone, female hormone, and long-term estrogen exposure can lead to dysplasia. Okay, long-term exposure of the estrogen leads to hyperplasia, and hyperplasia can eventually progress to dysplasia, and dysplasia can lead to cancer. It is for the endometrial hyperplasia. Okay, not all hyperplasias are cancerous. Not all exceptions are always there. For example, benign prostatic hyperplasia. It is a hyperplasia in the prostate glands of the men, and in which it do not increase the risk for the prostate cancer. It is an exception. While mostly hyperplasia can lead to cancer, but benign prostatic hyperplasia is an exception. This one was for hyperplasia and hypertrophy. Let's discuss about atrophy. Atrophy in which the when any stress is reduced to an organ, the size of the organ will also be reduced, and it can also occur in two ways. Number of cells can be reduced and the size of the cells can be reduced. Number of cells are reduced by apoptotic mechanisms, and the size of the cells is reduced by the autophagy and ubiquitin proteasome pathway. Okay, I will discuss them one by one. First of all is the ubiquitin proteasome degradation or pathway. Ubiquitin is a protein, and it is a protein present almost inside all cells, and its function is to protein clearance, is to maintain the protein clearance of the cell. For example, intermediate filaments of the cytoskeleton. Cytoskeleton are made up of filaments. One of them is intermediate. These are tagged with ubiquitin and are destroyed by proteasomes. Proteasomes are proteases. Proteases are enzymes that destroy the protein. I hope you understand the concept that ubiquitin tagged proteins are destroyed by proteasomes. For example, this is a protein which is to be cleared, and this needs to be cleared, and for example it is dangerous to the cell or in the atrophy something, so it is tagged with the ubiquitin protein, and this is identified by the proteasome. This is identified by proteasome, which is a protease. It is identified by proteasome and eventually it is degraded. It is known as the ubiquitin proteasome degradation. Autophagy is the self-eating. Auto means self and phagy means eating. It involves the generation of autophagic vacuoles, and these vacuoles fuse with the lysozymes. For example, it is a mitochondria, and it has to be destroyed. It encloses within the autophagic vacuole, and this vacuole combines with the lysosome. These are the hydrolytic enzymes within the lysosome, and they form phagolysosome. Okay, in which there is the organelle which is to be destroyed and hydrolytic enzymes, and eventually it causes the breakage of the mitochondria. The next thing here is the metaplasia. Metaplasia means that when the stress is changed, it leads to the change in the cell type. For example, squamous can be changed to cuboidal, cuboidal can be changed to columnar. There are three kinds of surface epithelium. Surface epithelium, the epithelium which lines the body. So these are squamous, Okay. There are three kinds of surface epithelium. First is the squamous, then columnar, and the urothelial. Urothelial, which lines the urinary bladder, and it is also known as transitional epithelium. The most common and famous example of metaplasia is Barrett esophagus. Barrett esophagus, it is in response to the GERD. GERD means a gastroesophageal reflux disease. It means that the stomach contents are refluxed back into the esophagus. As you know, that pH of the stomach is very low. It is acidic. It is highly acidic. So the cardiac sphincter protects it from reversing back. But there are some conditions in which the reflux can occur. So, the stomach is lined by the simple columnar epithelium, and the esophagus is lined with the non-keratinizing squamous epithelium. But here in Barrett esophagus, the cells of the esophagus undergoes metaplasia and they are converted into non-ciliated mucin producing columnar cells. Like they also produce mucus and they are non-ciliated and these are columnar. As you can see here, these are the squamous cells. Here, these are the squamous cells, and but this condition, here you can see that there is the metaplasia going on, in which the squamous cells are converted into columnar cells, and these are, and you can see here these are the mucin producing goblet cells. While usually the goblet cells are not characteristics for the esophagus, but in the Barrett esophagus, in case of GERD, they can be induced. Okay, but the metaplasia is reversible. I cannot stress it enough that metaplasia is reversible. For example, if the gastroesophageal reflux disease is reversed or it is treated, the cells can be reversed back into the squamous type which they were originally in. So, but the persistent stress, for example, the GERD is continuous and this is not treated, it leads to dysplasia and eventually it leads to adenocarcinoma of the esophagus. This is the progression and the prognosis is not good. It is severing the condition by not treating the underlying cause. Always there are exceptions. Metaplasia not always lead to cancer. There's an exception known as the apocrine metaplasia of breast. Apocrine metaplasia of the breast means that the breast epithelial cells are converted into apocrine or sweat gland type of cells, which is not the characteristics of the breast cells, but this is the metaplasia. Maybe some trauma, maybe some underlying cause induce them to convert it into sweat glands type, but this is not progressing into the cancer. This is an exception, apart from the adenocarcinoma of esophagus. Okay, other thing, vitamin A deficiency. Vitamin A is necessary for the differentiation. It is a vitamin and it is needs for the proper differentiation of the epithelial surfaces. For example, conjunctiva covering the eye. The conjunctiva has normally covering of the squamous cells, squamous epithelial cells. But in case of vitamin A deficiency, it can be metabolized into stratified keratinizing squamous epithelium. It is still squamous, but they are stratified, many layers. It means many layers of squamous, which is keratinizing epithelium. And in the image, this which I am highlighting in green represents the keratomalacia. It is a condition in which the thin squamous epithelium of conjunctiva is converted into stratified keratinizing squamous epithelium, and it is a disease which can be common in people who have undergone some kind of metaplasia. Other thing is the mesenchymal cells. Mesenchymal connective tissue are the connective tissue type, in which they form bone, cartilages, the circulatory system, blood vessels, etc. The metaplasia in these can cause myositis ossificans. For example, in this diagram, it is an X-ray induced diagram in which we can see that this is a long bone. It can be either humerus, it can be femur, it can be any of the long bone. But the beside the long bone, this is the muscle. But you can see here it is a slightly whitish appearance body. It is the myositis ossificans, in which the muscle tissue changes into the bone. There is the formation of bone inside the muscle, and the underlying cause can be trauma. Underlying cause can be any like stress condition, change in the stress, which induces metaplasia in the muscle. The last is the dysplasia. Dysplasia means disordered cellular growth. Disordered cellular growth, and dysplasia always leads to cancer, and it is reversible. Dysplasia is also reversible, but after it progresses into the carcinoma, it becomes irreversible. So, for example, cervical intraepithelial neoplasia, it represents a type of dysplasia, and it is a precursor for the cervical cancer. As you can see that dysplasia can lead to carcinoma or cancer. It arises from the pathological hyperplasia, which is long lasting, which is chronic, which is not treated, which um, which is very long standing. It leads to dysplasia. For example, endometrial hyperplasia and Barrett esophagus. If these two conditions are not treated, they can eventually lead to cancer. And CIN, cervical intraepithelial neoplasia, is an example in which it leads to dysplasia. And it can lead to carcinoma, and that condition is irreversible. And these four mechanisms are growth adaptations in the cell in response to stress, but the two other terms like aplasia and hypoplasia are also used. Aplasia means A means no and plasia means growth. A means the failure of the cell production in embryogenesis. For example, if any organ is not fully developed or not produced in the during the embryonic life, the example is the unilateral renal agenesis. It means that the baby is born only with one kidney and the other kidney is not present. Hypoplasia is the decrease in the cell production during embryogenesis. It leads to small organs. Hypoplasia, hypo means small, and it leads to small organs. For example, streak ovary in Turner syndrome. Turner syndrome is a congenital disorder in which the chromosomes number are fluctuated, so it leads to streak ovary. And this can be set as an example. So this one was for today, and I hope you understand the concept that hyperplasia, hypertrophy, metaplasia, dysplasia, atrophy, and hypoplasia, aplasia, and hypoplasia. These are the growth adaptations, and the cell undergoes these adaptations only in response to stress. It is not physiological apart from the like uterus development during the pregnancy. It is physiological, but most of them are pathological. So that was for today, and in the next, we will discuss the principles of the cellular injury. This topic, and okay, this was for today, and take care. Goodbye.